Okay, so let me see back a little bit. I'm doing a YouTube channel with my school project, and I thought it'd be super cute if you guys saw it. And it's over biology, so if you're in biology, I hope this helps. Um, I kind of messed up right here. I put my finger through it, but I'm going to be going over protein synthesis. Yay! Okay, so the first process, the first step is transcription, and that happens inside the nucleus. Um, so, as you can see, I kind of have this little space right here. That's where it used to, like, it's just a regular DNA strand, and then my RNA polymers, I can never say that right, but it goes through and kind of like unzips it. It's like it just unzips it, and you have your strand, mRNA, messenger RNA. Before we get any further, I should probably explain the difference. There's um, DNA and then there's RNA. DNA is deoxyribonucleic acid, and um, RNA is ribonucleic acid. So there's a difference. Um, Next up, after we go through transcription, oh, you should probably see this part too. By the way, this is a key down here. So, the blue is thymine, A is adenine, green is guanine, and red, or it looks pinkish, I guess, is cytosine, and then this purple right here is uracil. But you get to this when we talk about this. We're actually going to talk about this. So let's go into this. Drop it. Okay. Next up, we have translation. So, after we go through transcription, we're going to go into translation. And as you can see, we have our mRNA down here, this little strand right here, messenger RNA. I will explain that after. We have our codons, which are these three prongs, and I know they're not actually prongs, but I drew them to where they look like prongs, and it makes sense to me. So that's how I remember it. We have down here our codons, and up here our anticodons. So there's a difference. Our codons, excuse me, our codons, they're always in a sequence of 3. So as you can see here, we have uracil, and then this red one, which is a cytosine. And as you can see over here, my little example, I have 3 black ones, 3 black prongs, and black prongs is adenine. So normally in a DNA strand, you would pair it up with a thymine. But because it's RNA, we're going to pair it up with uracil. So then I have 3 purple little prongy thingies right here, and they connect, and yay, translation. So, another big thing to remember is, as you can see, it's like a blue little bubble thing. Ribosome. It's found inside the cytoplasm. So, all of this happens inside the cytoplasm. So, but all of this is a ribosome, and it consists of RNA and associated proteins. I don't know if I explained that right. And you're probably wondering, well, how do these little prongy thingies get to the other prongy thingies? tRNA. Okay, tRNA. Transfer RNA. That is when tRNA, transfer RNA, it's transferring amino acids. I will also talk about that later. So yeah, it's really important to know that that codons are a sequence of 3, anticodon sequence of 3, and they have to match up. So I am done with that. I'm going to go over the RNAs right now. So the RNAs, done with that. RNAs, so basically, mRNA, messenger RNA, right here, it carries the instructions from the DNA to the rest of the cell. So after we go through this whole process, it carries it out. We start over with RNA. tRNA, transfer RNA, transfers each amino acid to the ribosome, which is this. So once this gets to here, then this ribosome shifts over, and these two are already connected. They break off, if that makes any sense. The last RNA is the rRNA, lowercase r, capitalized RNA, ribosomal RNA, and it's basically just a bunch of proteins, a polypeptide, if you will. Excuse me, that should be right. So it's super important that you remember that proteins and polypeptides are made up of amino acids, and proteins are a chain of amino acids. That's super duper important. And something really cool that I learned is enzymes help DNA and protein synthesis by breaking hydrogen bonds between the bases and allowing strands to duplicate. So I hope you guys learned a lot. I probably just sped that up a little bit. And I know this is a totally random video and my lighting sucks. I'm super sorry guys, but I still thought it'd be super cute, and I hope you liked my little sciencey side of me. Yeah, thanks for watching guys.